Okay, so this is the microscope lab. This is part 3. This is where you make your own slide and look at it through the microscope and do some other stuff with it. So I've got a box of slides here. We reuse the slides and the cover slips over and over and over again because we are cheap. But also, anytime you get a slide, even a brand new slide out of a brand new box, you always want to clean it. So in this case, I'm just going to run some water on it. Water on. We're in the other lab, incidentally, for people that have been in my lab. The video equipment is in the microbiology lab. So I got the slide nice and wet. I'm pinching it with a paper towel to clean it off. Getting it dry, trying not to get too many fingerprints on it. And if I do, getting fingerprints just on the ends. So I got a slide. Now we also reuse the cover slips, which are a little trickier. Very, very thin pieces of glass. You probably can't even see that. But I'm also going to get this wet. And now to pinch it is really tricky because if you pinch too hard, it breaks the glass. The nice thing about cover slips is that they don't break sharp, so even if you break one, it's not going to cut you. But you do need to get them clean because if you're focused under the cover slip, you're focused on the cover slip too. And if there's dust or anything on the cover slip, you're going to see it. Okay, so my cover slip's nice and clean. And I just realized, because this is not my lab, that I don't have, water. Drop of water.
You gonna keep the sink going?
Thank you, director. That would have been distracting with it. So, drop of water. I always talk about a Goldilocks drop, not too big, not too small, but that doesn't make sense until later. So I'm gonna put a drop down. Now we are gonna look at our own cheek cells. So take a toothpick, take the tip of the toothpick on the inside of the cheek. I scrape, put the tip in the water because you want the cells to come off. You can't see them. Take the cover slip, put it on the drop. And now, to the microscope. Now, if you want to set the thing up for your elbows while I'm setting up the microscope, we did not do that.
Yeah, I'm okay.
Okay. Okay, this line that's on the screen right now is the edge of the cover slip. Because what's under here, there's a, let's see, I gotta look through the scope. There's an air bubble. Let me close the iris diaphragm. And get this in focus. So air bubbles, very recognizable edge. Sometimes it traps oils or other things from, the microscope slide was clean, but it's not as clean as it could be. That's where the streaks have come from. But at low power, cheek cells are visible, but they're not really, really visible. Okay, I think I'm looking. I'm going to move this to the side just a little bit. At low power, these little things are cheek cells. So they are visible as long as the iris diaphragm is closed, but you can't really see them terribly well. But I know where they are. And what I want to do is put the air bubble in the middle of the field, that edge in the middle of the field. So when I go to 10, there's that nice dark line, and that gives me something to focus on. Those cells that are right up against it are not great. There's a much better cell just off to the edge. You can't see it that well right now, but that's a much better cell. But we're not at the right power yet for drawing the picture. In your lab, it says to draw one cell at high power, and we are at middle power. But I want to focus with that bump right in the middle because I know that that cell that I want to get to is just off to the right of that edge. But when I go to high power, it's going to be off the edge of the field. So there's the edge. I'm going to get that nice and focused. And I'm going to travel out, and there's the cell. Actually, there's the cell. This is a pretty typical cheek cell, and that's what you're being asked to draw. So it's a little crumpled. Cheek cells are pretty flat, and they do crinkle up a little bit. You can see that these are really crinkled. But that is a nice one. Is it it could be a little bit rounder, but it's a pretty decent cheek cell. And in terms of using the microscope, this is by far the toughest process thing that you would have done in the lab today, is to get this so that you could find a nice cheek cell to draw. Now, if I can get my videographer to come over here, the next thing we're going to do is we're going to stain these cells right where they are. So I'm going to go down to middle power. It gives me a little bit of room to work. I've got a bottle of Hucker's Crystal Violet stain. This is actually a stain for bacteria, but it stains really fast and really reliably. So for this lab, it works pretty well. So I'm going to take and put a drop right alongside the cover slip, and it should bleed underneath. This is why you don't want too big or too small a drop. And if you go back over here, as it moves, it's pushing cells along. But it is gradually coming into the field. See, there's a stained cell going by. You can see the edge of the stain coming underneath. Is that the stain will eventually start to stain here. The cells will pick up the stain faster than the background. I'm going to move over to this clump of cells over here. And if this works the way that it's supposed to, and it's biology, so that's never a sure thing, they should start to pick the stain up as it comes across. If you'd started with too big a drop, the cover slip would have been surrounded by water and the stain would mostly have gone around the cover slip. Too small a drop, and for most of you, by the time you found your cheek cells, that drop would have evaporated away and there wouldn't have been any space for the stain to go underneath. You know, I've done this a few times, so I could do it pretty fast. But you can see the stain starting to interact with the biological material in these cells. And your next question is, describe what happens as the stain reaches the cells. And so you're actually watching the stain interact with the cells. You also have the benefit of having this done by somebody who's done this a few times. Because a lot of times when you do this on your own, by the time you look back through the scope, it's already done whatever it was going to do. We're right on the edge, that the edge of the stain, my little drop is completely depleted. So the stain isn't really coming much farther across, so you get a chance to very slow motion-y see how it interacts with the cells. Now, I've mentioned that the labs have tough questions on them. The labs also typically have duh questions on them. So your next two questions are duh questions. Don't sweat them. Is that the next question is, I've forgotten what the next question is. I know the next two are kind of dumb. Oh, if you'd put this, how do the cells look different now than they did before? That's number 3. And that's pretty easy to do. And number 4, if the first thing we had done before we went looking for them was to put the stain on, how much easier would it have been to find them than to do it without the stain? And that's, you know, that's a dog question. Okay, that is part 3. We have one more part to go. For some labs, there's a part 4 and 5, and for some labs, the part 4s are different. So different part 4s and 5s will be up for different labs doing different classes. But we're going to record part 4 for SC135, or SC137 actually. That's what I'm doing, human biology. Okay, we can stop there.